Thank you, Guppy, and hello to everyone. I'm coming to you from the third floor of the Hutchinson building in my office. And as Guppy mentioned, I'm a scientist here in the Division of Human Biology, but I also have another role as the Senior Vice President leading the Office of Education and Training at Fred Hutch. And so many people don't realize how much training that we do at Fred Hutch, but we have everything from programs where we work with high school and middle school teachers on curriculum development to internships for high school students and undergraduates. We have about 150 graduate students training at Fred Hutch, another 200 postdocs, and we also help support the professional development of faculty as they get started in developing their labs. So so before I introduce my fellow panelists, I'd like to share a little bit about Fred Hutch. So for those of you who are newer to us, Fred Hutch is an independent nonprofit organization and the only National Cancer Institute designated comprehensive cancer center in the state of Washington. Our researchers are committed to transforming the way we understand and treat cancer and infectious diseases by taking science from concept to cures. This year, we're marking Fred Hutch's 50th anniversary and the incredible progress that we've made since opening our doors in 1975. So scientific breakthroughs don't happen overnight. They come from persistence, mentorship, and institutional stability, which gives great research teams the support to both be nimble in response to emerging opportunities and relentless in the pursuit of ideas that may take years to mature. That stability has been supported over the years by donors, including many of you in the audience today. So thank you. Your contributions make you part of our ambitious campaign for Fred Hutch and truly help accelerate and expand our work. Another important source of funding is the federal government, including the National Institutes of Health, or NIH as we often abbreviate it. While many think about funding from the NIH as being used to provide the materials to do research, these funds also provide salary support for trainees who are the ones that do much of the day-to-day work. The NIH also provides numerous grants directly to trainees to support their development as they work towards careers in science and research. That means changes in federal funding priorities and mechanisms affect trainees as well as the progress we can make now and well into the future. So to help me talk about the scientific training pipeline and some of the effects from the shifts we're seeing in the funding landscape, I want to welcome Chris Peterson and Kelsey Woodruff. So Dr. Chris Peterson came to Fred Hutch as a trainee. He was a postdoctoral fellow in the lab of Dr. Hans-Peter Kim, working on cell and gene therapies. His research has focused on making better immunotherapies against cancer and infectious disease. And he was so outstanding that Fred Hutch hired him as a faculty member a few years ago. So he's just been setting up his lab. Kelsey Woodruff is a graduate student getting her PhD in the lab of Dr. Tina Termini. Kelsey is studying the importance of certain molecules in the development of acute myeloid leukemia, a blood cancer, and she recently received the Dr. Paul Neiman Outstanding Graduate Student Award, which celebrates exceptional students in the Molecular and Cell Biology Graduate Program, which is jointly administered by Fred Hutch and the University of Washington. Welcome to you both.
Thanks for having us. Great to be here.
Yeah, thanks, Nina.
Excellent. So to get us started, can each of you share your journey in science and how you got where you are? And maybe we can start with Kelsey.
Yeah, sure. So when I started my undergrad originally, I was on a pre-med track, so I wanted to go to medical school. And towards that development, I started doing undergraduate research with PI at my undergrad institution, Ball State University. And I think that was a really transformative experience for me. I think my PI was really committed to trainee development and career development. And as an undergrad, I actually had the opportunity to get some fellowships from my university that paid me to do research, which is really uncommon. But it was a big help in really allowing me that time to focus and like develop my scientific understanding and, commitment to research. And so when I was a senior in my undergrad, I applied to PhD programs, and I was very fortunate to be accepted to the University of Washington and Fred Hutch. And I've been here for about 3 years now working on my project. So.
Excellent. I just want to pick up on one thing that you said, Kelsey. You mentioned that that actually getting paid to do research was really helpful, and I think maybe could you expand on that a little bit more and about sort of who has the opportunities to be able to engage in research while trying to get undergrad and get that going.
Sure. Yeah. So typically for undergraduates, I would say those research opportunities are like an unpaid internship model, which works well for some people. But from my situation, I didn't really have financial support from my family. So it was really like on me throughout college to make money and like pay my bills. And so having the opportunity to have like a fellowship instead of, having to be a barista and then squeeze in lab work between my classes and those types of shifts really, I think, let me focus on what I wanted to do. And I think like I was really able to excel in that environment because I didn't have the extra financial constraint, so yeah, it was really impactful and important for me, I think. Yeah.
And I think I just for the audience's benefit, want to say that, Kelsey was one model where she was able to get some research experience during undergrad and apply directly to graduate school. But actually, one of the newer programs that we have at Fred Hutch is a post-baccalaureate program where we give folks that maybe didn't have the opportunity to do research while they were an undergraduate because they were focusing on their studies or maybe shifted their interests over time and had to take other classes, or as Kelsey mentioned, just work to be able to pay the bills, that after they graduate they can come to the center for a paid technician position but also get professional development towards getting them ready to be able to apply towards graduate school. And I see Guppy has nicely put this pipeline showing the sort of different phases that the Office of Education and Training supports at Fred Hutch. So maybe next we can shift to Chris and hear a little bit about your journey.
Yeah, so my journey's been a few extra years relative to Kelsey. So I graduated, I got my undergraduate degree from University of North Carolina in Chapel Hill in 2002. I also went directly from undergrad to grad school. The graduate stipend, as you all are highlighting, was a big factor in me making that decision. As Kelsey said, I was interested in a graduate program. I'm also a musician, so I was actually looking at a lot of different options. And as opposed to being a professional musician, there was a lot more stability that was provided to me, even by having a small graduate stipend. So that was really the factor that was the tiebreaker for me. So I got into a few graduate programs. The one that was the best fit for me was the University of Utah. So I moved all the way from Central North Carolina to Salt Lake City in 2002. Um, I don't think that I would be able to do what I did then today. It's so much more of a competitive atmosphere. I had a lot of lab experience as an undergraduate, but I didn't have publications to my name. I sort of was helping other people with their experiments rather than showing my own sort of independent thought. And that's a really important part these days of getting into these graduate programs. So the post-bac programs that Guppy and Nina are highlighting, I think, are really important for where the field is today. So I finished my PhD. I finished my PhD as the financial crisis was hitting in the late 2000s, and that was its own interesting experience. My first postdoc, they were not sure they were going to take me because they also needed to buy a freezer. So they were deciding between me and a freezer, but they happened to get supplemental government funding, and that allowed them to buy their freezer and also hire me. And again, that was a really fortuitous aspect for me, highlighting all, the funding that we're talking about today is going to help more people really follow the path that I followed. I joined Hans Peter Kiem's lab here at the Fred Hutch, and I sort of rose through the ranks internally over the next 12 years and opened my lab here in 2023.
Excellent. And that's very interesting that you sort of came forward in another challenging financial time like we are in here. And I mean, it is true that these things are cyclical. I remember a point in my career where I had been here for about 5 years or maybe almost 6 years, and I was right at the cusp of promotion from assistant to associate. And one of the things that you need to do to demonstrate for that promotion is that you're really you've gotten yourself going, you're advancing the science in new ways. And I was super excited about a new project that had been developed by a graduate student. So it turned out, Guppy mentioned that I work on a bacterium called Helicobacter pylori, which infects the stomach and is the biggest risk factor for stomach cancer. But evident in its name, the shape of the bacterium actually is a helix. And we had some kind of indirect evidence that actually cell shape might be important for this bacteria to colonize in the stomach. And I had a graduate student that really was pushing that forward. And she had enlisted the help of an undergrad to help her do a screen to get mutants that were altered in this helical shape. And we were actually collaborating with a small biotech company up in Lake City that had this fancy kind of fax machine that had a special objective on it so it could sort really small things like these tiny bacteria that we worked on. Anyway, this was like a super cool project. I was super excited about it. I had put in a grant on this sort of new research direction, and it didn't get a very good score. And then this high school student wanted to go to the American Society of Microbiology meeting because she had gotten really excited about research and she wanted to use that as an opportunity. The meeting was in Philadelphia that year to talk to people and look at schools in Philadelphia. And I said, of course. But then that funding didn't come through. And I was like, oh my gosh, I'm going to have to put this on my credit card because I cannot say no to this great enthusiastic student. And then that was actually right when the American Recovery Act kicked in, and they had given money to NIH, and different institutes did different things with that money. But the National Institutes of Allergy and Infectious Diseases used that extra money to fund a lot of R21s, which are exploratory 2-year grants. And that was the grant that I had written, was an R21. And so it got funded on May 15th, and the meeting was like May 20th. So the student got to go. I didn't have to pay for it off my credit card. It was a happy ending. But that's just kind of, here again, an example of, kind of the craziness that, faculty are, in a sense, these small business owners, and we depend on the enthusiasm and the drive of trainees to advance our science. And there's so many examples of that I could give. Actually, why don't we have Kelsey and Chris share some of the exciting discoveries that they know about from their own work or trainees that they've been around. Chris, do you want to start us off?
Yeah, so I've had or mentored a couple graduate PhD students. One, I started a mentorship when I was still in Dr. Keem's lab as sort of a senior scientist, and then when I started my own lab, he and I co-mentored this person. Her name is Lucy Maynard. She had a really amazing project. She came to us from the bioengineering program at University of Washington, and she was very good at designing protein molecules. And we design protein molecules that can detect cells that are infected with HIV, and also cells that are malignant and would contribute to cancer. And so Lucy designed a series of 20 of these anti-cancer molecules. And then we tested them in an animal model, and we were able to just basically pick the winner. So we put them all in and tested which one worked the best, and then used different assays to rank order them. It was a really amazing project. It's not something that can be done in clinical studies, so it's a good example of our preclinical research that's so important to fund. Lucy finished her PhD in under 5 years, which is really exemplary, and she got a job in the biotech sector immediately after she graduated, which is also very rare, especially these days. So she was really an amazing PhD student. I have one now that's just started named Lydia Gagarina, who came to us from Russia. She's really been an amazing addition to the lab, a lot of new energy, and she's working on a project that will allow us to deliver those anti-cancer and anti-HIV molecules to the body without all the expense that's really associated with some gene therapy. So Lydia is a good example of someone that just brings sort of new life and new blood to the lab, coming from a different perspective and having a lot of energy that's infectious, for lack of a better word. So the contribution of these trainees to my lab has really been indispensable.
Excellent. Thank you for sharing that, Chris. And there's a question in the chat about, do Fred Hutch and University of Washington share graduate students? And the answer is yes, at multiple levels. So Kelsey is part of the MCB Molecular and Cell Biology graduate program that we actually jointly run with University of Washington. But we also have students from more than 15 other departmental and interdisciplinary graduate programs, such as the program in bioengineering and many other programs. So that's how come we have over 150 graduate students training at Fred Hutch at any one time. But let's hear from you, Kelsey. Let's share some of the things that you've seen and got you excited.
Yeah. So I guess I can share a little bit about my lab and the work we do. So kind of like Chris, we're a newer lab. I think Tina maybe started a few months before you did, or maybe a year. I don't remember quite. But I was the first graduate student in the lab, which is kind of a challenging position, but it's also a really exciting position because I think I've really had the opportunity to drive a lot of, certainly drive my project forward, and then also make contribution to the work of others. But, I think Nina mentioned at the beginning, I study how molecules on the surface of leukemia cells impact cancer progression and chemotherapy resistance and survival and things like that. And so what I've found over the past 3 years of being in the lab is that actually if you change one of these molecules in a really particular way, like very, very tiny change, and the cancer becomes way more susceptible to chemotherapy. So we're really hoping to continue interrogating that further and maybe develop new biomarkers or a new way to target these molecular interactions to really improve outcomes for leukemia patients. But yeah.
Hey Kelsey, there's a question in the chat for you about what stood out about Fred Hutch that made you want to join this program.
Yeah, that's a great question. I think the thing that really jumped out to me about Fred Hutch is just the amount of collaboration that exists within this institution. And I think Fred Hutch really has a commitment to making science easier to do, which is not always the same at different institutions. But we have really excellent shared resources facilities here. And I've been able to work with most of them throughout my PhD. So it really has enabled me to answer questions about my project that I just wouldn't really have had the expertise to do otherwise. But because those people are here, it's really easy to do that work. But,
yeah. I love that you highlighted sort of this very collaborative nature and also the impact of our shared resources. So to explain what that is, to people in our audience, to do our work, it's very high tech. And so there are numerous super expensive instruments that help us analyze cells and what they're expressing and be able to sort them. And for using DNA technologies to be able to sequence very deeply to understand the genomes as well as what genes are actually being expressed in different cells and under different perturbations. And so there's and then looking at the proteins, you name it. And so we have these excellent resources where they're run by PhD level scientists, but they're not running a research program. They're devoted to these instrumentation and allowing our researchers to use them. And this has been a big part of the Hutch since day one. And in fact, many of our faculty collaborate with these shared resources directors to actually develop new technologies that allow us to answer new questions. And, and so like, and a lot of the times the people here again that are actually doing the work are actually, the trainees. So like, trainees in Jesse Bloom's lab, developed new methods to do viral forecasting based on a new method of doing deep mutational scanning and figuring out, where mutations could happen, and then, then actually looking in data sets and seeing like, yep, look at this, we're getting, mutations in exactly where we predicted. So it's a really, I think it's a great feature of Fred Hutch, and trainees notice, when I was co-director of the MCB program for 6 years, and we would do our spiel during recruitment, these were the things that we would highlight to get students excited about coming to our program. But maybe we can have a little bit of a transition now to talk about that elephant in the room, funding. Chris, can you share with us how trainees in your lab have been funded?
Yeah, so when I was promoted just over 2 years ago now, it had followed 12 years in my postdoctoral fellowship lab, and I had a number of titles after postdoctoral fellow. That's a very long time, but the reason that it was such a long time is that it's a longer path when you want to be promoted internally. I probably could have looked for faculty jobs elsewhere within the first 5 to 7 years of my time in Dr. Keem's lab, but I really love Seattle and I wanted to stay here, and I was just patient to sort of keep doing the work that I was doing. I was doing what I enjoyed doing and to see where it went. So over those 12 years, I was able to build up through my publications and all of my research 3 NIH grants. So when I was promoted, I had 2 R01 grants, which R01s are sort of the main 5-year grant funding mechanism that we all shoot for as biomedical researchers. And I also had a program project, meaning that it's a grant that funds me here at the Fred Hutch, and then we work with people at the University of Pennsylvania and at the National Institutes of Health and elsewhere, and we all do collaborative science. So I was very fortunate in that when my lab started, I already had funding from the get-go, and that's the funding that supported Lucy and Lydia, as well as some postdoctoral fellows that are in my lab. Now, if I had gone elsewhere, if I had left the Hutch after 5 to 7 years and gone to the East Coast or California or wherever, I wouldn't have had that money. And that leaves people in a much tighter situation. So the clock is really on. You're given some funding to sort of get you on your feet, and that's great. It's helped me as well. But the fact that I had the additional funds really made the difference. And going back to what we're talking about today, I think that's what we want to provide to these labs and these trainees is the stability to do the research that they need to do.
Yeah, my experience was sort of more like the kind of latter scenario that Chris talked about. I came here, and I'd had fellowships both in graduate school and as a postdoc from the National Science Foundation in graduate school, and then I had a private grant from the Jane Coffin Child Trust for Cancer Research as a postdoc. But then I came here and I had a little bit of startup for a couple years, and then I just started recruiting people. And my first graduate student was actually from Venezuela, and I didn't realize that since she was not a U.S. citizen or a green card holder, she could not be on any of the training grants that we have that students and postdocs can apply to help support them. So, but luckily, I got a new investigator award from the Pew Charitable Trust. They have a biomedical scholars program. And that money, it wasn't very much. It was, I think, $60K a year, but it was unrestricted. So I actually used that money to help support Dahlia, and then, started to be more aware of, of the restrictions. And I think there's a lot of this sort of learning by doing on the job. But then, that brings up another challenge. And over the years, my trainees have been pretty successful on getting onto various training grants. But one of the additional challenges is that Seattle is an expensive place to live. And so stipends are actually larger than what is allowed on these NIH training grants. And so we have to supplement those funding sources. And there's restrictions on being able to do that from federal grants. And so, here again, that sort of leads into the kind of trickiness. One way that you can help support students, and particularly for new opportunities, is supplements, which I think Chris or somebody had, or maybe it was Kelsey, had mentioned being supported by a supplement at some point. I had a supplement for a super cool project that was also directed at bringing people that have not historically had a lot of participation in science into our laboratories. And so I actually had a Native American student that worked on this, that was interested in this project, where we were finding that Helicobacter pylori strains that infected certain populations had a different complement of virulence genes, and she wanted to really dive into that. And so that was really exciting that we got the supplement, but then that supplement was canceled basically in February of this year. So suddenly we were left, scrambling. Luckily for me, I actually have an endowed chair. So I hold the Penny Peterson Memorial Endowed Chair in Lymphoma Research, because Helicobacter, it turns out, causes a rare lymphoma in the stomach. And so luckily I had some of those extra funds that I could, scramble to continue to support this work going forward. But it's, here again, lots of challenges, lots of different ways that we support trainees. Anybody else have anything they'd like to add to that?
Sure.
Okay.
I mean, I can share a little bit about how I'm funded as well. Yeah, because I think so. Right now, I have a graduate research fellowship from the NSF, so not the NIH, but another national institute that's kind of like facing similar problems. And so I'm lucky that my award is safe and the funds are coming through right now, but this year they've actually restricted who's eligible to apply for that award in such a way that like I would not have been eligible to apply at the stage that I did. So I think it's really kind of limiting opportunities for those training grants and for those fellowships for graduate students that PIs really depend on, like Nina was saying, for to
yeah, and just to give a little context, because I also had an NSF and back in the day. So the National Science Foundation has historically given graduate research fellowships to students across the nation in a wide range of disciplines. So biology, but also, you know, engineering and physics and chemistry. So they have a very broad portfolio. And, you know, in recent years, they've given about 2,000 of these fellowships, and you can apply before you start graduate school. And then it had been that you could apply also in your first and second year of graduate school, because depending on the type of program you're in, sometimes you go directly join a lab, but actually in our programs in the biomedical sciences, actually during the first year of the program, the students rotate in 3 different laboratories over the 3 quarters of the academic year. And only at the end of the spring, so at the beginning of the summer, do they actually join a lab. And so for those students, they actually, if they're going to apply to the NSF, apply in the fall of their second year, because now they have an actual project that they can describe, as opposed to just like saying, this is what I vaguely might want to do. So in the past, like I said, they've given about 2,000 of these. This last year, they actually only gave 1,000, so half of the grants. And then for this year's call, they've added this restriction where they're basically not allowing second year graduate students to apply. So you have to apply either before you start graduate school or within that first year.
And I would just follow up by saying, you know, I think the 2 of you that received that NSF award, I applied for one as well. I did not get it when I was in grad school, so it's an incredibly prestigious honor. But it just highlights that that's only a small percentage of people that are getting into science that can really avail themselves of that, you know, a thousand or two. And there are more people that are entering science, and I think it's up to us to support them. I think we need more great minds in the scientific realm. The current year is going to be challenging for new graduate students, and we want that to be the exception rather than the rule.
Yeah. I noticed that there is a question in the chat about what is an endowed chair. So this is a really great thing, and it's actually a program that has really grown in the last several years. Guppy can correct me if I'm wrong on this, but this was an effort that was initiated really by our prior president, Dr. Gary Gilliland, who at the time, I think when he started, there was maybe like 2 endowed chairs, and he was like, we need to grow this to, I think we're trying to get up to 40, but I'm not, like, here again, I'm going to ask the philanthropy people to correct me. But basically this is somebody that makes a donation that that then gives an annual amount of unrestricted funds to the chair holder, so that they can have flexible money to you know, say, support a trainee or start a new project in a particular direction. And, you know, some people have them where they're focused on a particular topic, like, you know, mine has to be related, or, you know, should be related to lymphoma research. Some have, you know, some are focused on, like the Innovator Network has one that's for junior faculty. And so it's not necessarily the topic, but the type of person. So, so these are really, really, really great sources of flexible, unrestricted funds that you can be very nimble and put where the need is, and also take some bets, you know, take some risks, because as funding gets tighter and tighter from particularly governmental sources, the way that these grants go is that there is a peer review stage, and then once where grants get ranked, and then they go to the programs, and in that peer review phase, as funding gets tighter and tighter, the evaluators get more and more conservative about what they're willing to fund, because they know that they're basically prioritizing taxpayers' money, and they want to ensure that the public gets their money's worth, and so they're going to bet more on sure things rather than these out of the box ideas that end up not maybe having impact right away, but later do. Like, I think a great example is actually one of our former Fred Hutch trainees, Craig Mello, who won a Nobel Prize for the discovery of this, RNA silencing, which was, initially described in this flatworm, C. elegans, and it was, a regulatory process, but then it's turned into an incredibly powerful biotechnology tool that Craig has gone on to study and start companies for. So anyway, yes, I see we've got a slide that gives a little bit more detail on that. Excellent. Well, I realize that we are kind of at that 35-minute point, Guppy, where you told us that we should be opening things up to questions. Should we get some questions from our audience? I don't want to feel like we're driving the whole conversation. All right. Yes. Shall I speak about the Next Generation Fund? Yes. So one of the ways that the Fred Hutch community is coming together to help young scientists get through these precarious funding landscapes that we're in right now is through the NextGen Science Innovators Fund. So we are trying to raise $2 million for this fund to provide bridge funding for trainees that have been affected by federal grants that have been discontinued, delayed, or suspended. And some of you attending today have already contributed to this fund, and so we want to thank you very much for your generosity. So, the reason that me and some other colleagues really advocated to develop this is that trainees are such a powerful way to keep the scientific pipeline moving, to advance research programs, and also to preserve the future of new breakthroughs and cures, because, it really is, the trainees, I'm spend most of my time nowadays at the computer rather than at the bench doing the actual science, and they're the eyes and ears, and observing things that are like a little bit weird, and like, I wonder if that could mean something, doing their night science, as we call it, the project that you don't tell your advisor about, that you're just doing on the side. And that's really what drives research. And so we really feel that it is so important to preserve our ability to support trainees here at the center.
Yeah, I guess while we're waiting for more questions, I wanted to ask you, Kelsey, what are your next steps? What are you thinking about doing at the next stage of your career?
Yeah, I've kind of played around with it throughout my training. So I think initially I was really interested in taking a more like science writing or communication type of thing. But I think, I've really been able to grow my skills and like confidence at the bench throughout graduate school. And so like now I'm more interested in pursuing a postdoctoral fellowship and maybe being a faculty member, but I don't know. All of the uncertainty makes it, you know, you can be really excited about it and you have to be realistic. You know, if like these grants continue to be so competitive and like get delayed, like it makes it a little bit less appealing, I think. But I don't know.
I think as scientists we're all practical, so it's hard to think about
Yeah.
the higher endeavor, but you come out on the other side and you're happy you did it.
Yeah, it's a tough balance, you know, like being practical, but like, you know, still pressing on. Yeah. There is a question that about what kind of outreach we do with high school students to encourage careers in science, and we actually do quite a lot in this area, and I'm actually really proud of what we do. So I think I mentioned up at the top the science education, I didn't call it by name, the Science Education Partnership, where we actually work with teachers to develop science curriculum and also develop these kits that they can take back to their schools to actually implement curriculum. And so, you know, to help teach and build awareness, and you know, in particular, building modules around cancer biology, which isn't always a major focus in high school biology and various curriculum. Another thing that we do is we have a summer high school internship program where we bring students for a summer experience where they get training and also in pairs get to work in labs. But then we also have what we kind of call more exposure programs, which are, because the high school program is more for like raising seniors, but also like ability to reach students earlier and then also maybe after high school before they get into college, to have, you know, shorter programs where they can learn, say, about our shared resources, and also learn about the diversity of careers that make a cancer center work. So it's not just the, you know, the work in the labs, but it's also the work in the shared resources. It's also work in philanthropy. It's also work in facilities. And then there's on the clinical side of things, clinical research coordinators. So we have several programs that kind of introduce students to not only the research side of what it takes to run a cancer center, but also some of the other very varied careers that are possible, that are related to doing biomedical research. Okay, I'm seeing something, how NIH funding for faculty has changed since you started, and has it kept up with inflation? That's an excellent question. So it's actually really interesting because I joined the center in 2001, and I started, like, by the time when I got here, I felt like there was like a one piece of critical data that I needed to get before I could put in my first application. So like I got the lab set up, we got it going, and then I started in September, and so I put my first application in June of 2022. And it didn't get funded on the first try, but I got a decent score, and then I resubmitted it. And at that time, we were just coming off of the new Gingrich promoted doubling of the NIH budget. And so the pay line was the 18th percentile, and my grant had gotten a 12th percentile. Fast forward to that period I was telling you where I was like trying to, you know, renew my first grants and start to get some new grants. You know, financial situation had changed, and the funding line was 6th percentile. So that made it a lot harder. Another thing is, you know, for these R01 type grants that Chris mentioned earlier, which is kind of the bread and butter of research labs like ours, there was a modular and a limit, you know, where you could ask for modules of $25,000, and the cap on that was 250 or 5 modules. That has not changed the whole time. That's still what it is. And there is an opportunity to go non-modular where you have to, so for the modular, you don't really have to give much justification. But if you go non-modular, you have to really say what every dollar is going to be for, personnel, supplies, shared resources, you have to really spell everything out, purchasing the animals, housing the animals for the experiment, et cetera, et cetera. The cap for that has been and continues to be 500K. So that's it for the grant. And then superimposed upon that, you have things like, some institutes have limited the number of applications that you can have. So it's not like you can just write more grants. And also the expectation of the amount of science that you do as things get more and more competitive gets higher too. So that makes things very, very tricky. So things really have stayed flat. And then, since we've been on a continuing resolution now for, all of this year so far and continuing to next year, we're still at FY24 budget for the NIH. So that by definition, because of inflation and prior commitments, means that less grants are getting funded. Okay.
Somebody had asked previously what percentage of NIH grants get funded. Do you want to address that while we're on the topic?
Yeah. So it is not quite as bad as that 6th percentile that I mentioned. So that is a thing where if your grant gets a score and it gets this percentile, some institutes, and not all, will basically say, like, if your grant gets this percentile or better, it will for sure be funded, and then maybe it might be, they might go, depending on the scientific priorities, go to lower grants. If you actually look at like what percentage of people that put in applications actually get funding, it had been in the sort of 20-ish, between 20 and 30%, but it is tracking down. I actually don't know what the most accurate number is right now, partly because I think we're not collecting data right now. But yeah, so that's kind of where we're at. I'm seeing, is AI used in your research, and it is approved? It's interesting. I think AI is used in research in a number of different ways. I think many of the programs, for example, for analyzing DNA and things like that, have actually for quite a long time been using some elements of machine learning to help us analyze very large data sets. An area in my research where definitely AI has been helpful and is continuing to be helpful is in image processing. So when we take images of tissues and trying to define where are the individual cells and quantify the amount of signal of a particular protein that we're interested in, those softwares for being able to help us segment our images and figure that out, that is an area. Interestingly, there are newer and newer applications that are coming online. Even like to do things like help you design experiments more efficiently. And I would say that some of our faculty are actually driving that kind of work. I'm thinking of Sanjay in the basic sciences division, but and then there's also this really great initiative called Kaya, which is cancer AI. Ooh, what are all the letters for? But this is basically a collaboration between Fred Hutch and several other cancer centers, and several companies, including Cancer Center AI Alliance. Thank you. Like Google and Amazon and Microsoft, to actually have some test cases to see how AI can advance cancer research specifically.
Hi, Nina. Hi, Kelsey. Hi, Chris. Thank you so much for all that you've been talking about right now. But I want to jump back in with a couple of questions that are left unanswered from others in the chat. One of the questions that I think would be really interesting to discuss is, do you know of any trainees at Fred Hutch who have lost their funding and what their projects were by any chance? So Kelsey, I look at you first, possibly.
Yeah, I don't know anybody who's lost a training grant specifically, but I definitely know of one lab in the human biology division that lost about half their funding. So they study cancer metabolism, or last I heard, they had lost half their funding. So they study cancer metabolism, and that's really been impacting their ability to take new students, to complete the work that they've already started in that space. And yeah, and I think Nina mentioned that she had a trainee that had lost a supplement grant,
Yeah.
right?
And so I can speak to a little bit about this was actually kind of a tragic thing that was sort of on again, off again, on again, off again. Fred Hutch actually has a partnership with New Mexico State University, and that is a specific mechanism that basically got kind of caught up in the DEI language purge. And this lab had a collaborative grant, studying cancer metabolism with folks at New Mexico State University. And so that grant got pulled. And there are definitely other examples. So, I mentioned this diversity supplement, and, that was for Shiny Little Sun's project, where she was looking at this, it was actually also, it just so happened that project actually also had a health disparities aspect to it, because this is from a project that we're doing collaboratively with Duke University, where we've been really interested in the fact that Blacks have a 2 times higher rate of getting gastric cancer, and then also, if they have gastric cancer, of dying from their cancer. And so trying to understand what's going on there, what's the underlying biology, socioeconomic, is it just diagnosis, or how people are being treated, what treatments they're being offered? There's so many possibilities for what could be driving that. And I'm the Helicobacter pylori expert, which is the major risk factor for getting gastric cancer. And actually, very much to my surprise, I thought this was going to be a control. We actually saw a signal in the carriage of a particular virulence gene. And then we also had some hints that the actual sequence of that gene was weird. And so that was what Shiny's project was to dive into the molecular details of what's weird about this sequence. So anyway, those are just, a couple examples that I can give where, it's really interesting science is caught in the crosshairs of sort of what is politically acceptable right now, which we're not used to, I have to say, as scientists, about worrying about that. And so, I just put in a renewal of my H. pylori genetics grant. And, one of the things that's so interesting about H. pylori, which I just mentioned, is that strains that we collect across the globe have an incredible amount of genetic variation, which we usually refer to as genetic diversity. So I had to as I was writing my grant, I'm like, geez, I have to pull out diversity and make sure I say variation instead of diversity, because I don't want my grant to get flagged. So these are some of the challenges that the current political moment has placed upon science.
Yeah, and some of the other stories that I've heard, Nina, are also people who have been working in microbiome and had to remove the word diversity for similar reasons. Um, are there any divisions or research that are particularly at risk due to funding cuts that have more trainees, like more fundamental research?
Another area that has been particularly hard hit was the vaccine and infectious diseases division, because for a couple of different reasons. A, because vaccines became a verboten word. They also is the home of the HIV Vaccine Trials Network, and a lot of that work is global, and so a lot of grants got shut down if they had an international component to them. Luckily, in that case, most of that funding has been restored. Another area has been health disparities research, like I kind of just mentioned. I mean, luckily, the main grant that we have with Duke that is working on this cohort was not cut, but there have been other grants that have been cut, even though, you know, I mean, there's a lot of mixed messages that have gone out there. So I mean, health disparities work is not, you know, apparently inherently prohibited, but it's been a little bit, I would say, haphazard perhaps, in which grants get canceled and don't. You know, another thing that is kind of crazy is there's a lot of antagonism around mRNA vaccines and anything related to mRNA, but mRNA stands for messenger RNA, and it is basically, it is part of the fundamental way in which information in our genome is transferred. So in every single cell, there's mRNA. And I would venture to guess that almost like, you know, like most of the research that is done at the center in some ways does take into account mRNA. So it's like, it's kind of, you know, yeah, it's a bit of a crazy thing.
It really is, Nina. And what's really crazy is there was a recent study that it was not done at Fred Hutch, but Fred Hutch faculty member Josh Veach commented on it about mRNA vaccines actually improving outcomes for patients receiving lung cancer or melanoma treatment with immune checkpoint inhibitors. So it's a really fascinating, exciting piece of research, and that you know, thinking on a hopeful lens and finishing with another question in the chat. Starting with Chris, like, what's a discovery that excites you that you're working on? Is there something on the horizon? So Chris, Kelsey, Nina, something that is exciting for you.
I have the great honor to work with Dr. Drew Weissman, who won the Nobel Prize for mRNA vaccine research a few years ago. And I think the mRNA platform for me is the most exciting, because I think we can do things beyond vaccines, which in itself is amazing. But the way I use it is to make immune cells better at finding diseased cells, whether that be cancer or infectious diseases like HIV. So I'm really putting a lot of my focus and funding into that area. I think there's a lot of untapped potential.
Thank you, Chris. Kelsey.
Yeah, so I study sugars at the cell surface, and they're made by different genes expressed in your cells. And recently, I've actually found that when you introduce an oncogene, so a gene that gives you an increased risk for cancer, into a blood cell, they actually completely reprogram how much of these enzymes that they make. And so I think that's kind of like changing the sugar landscape and the signaling landscape for these cells. And so I'm really excited to kind of tease apart what that oncogene is doing and how those things are intertwined. Yeah.
That's super cool. Nina, closing project from you?
Yeah, you know, one of the things that I'm most excited about actually is this collaboration that we have with Duke University. And part of what's so exciting about it is that we're working with so I'm the microbiologist over here at Fred Hutch, and then the two researchers at Duke are a gastroenterologist and an epidemiologist. So we're being able to look at the question very holistically. But the gastroenterologist is basically collecting both stomach tissues and then gastric brushes that we can use to culture. So, you know, a lot of the studies that we've done is like, you know, from, you know, strain X, and then we use cancer cell line, you know, Y, that came from completely different, you know, places and medical conditions. But with this project, we're basically able to have the bacterial side of it as well as the tissue side of it from one person and understand that interaction at a new level. And there's like new technologies called single cell sequencing and things where we can really dissect things at a new level. So I feel like, I mean, this is the thing that's frustrating to me is like, I feel like right now is like one of the best times to be able to do science because we have new ways to collect a lot of data. We have new things like AI that helps us deal with these data. And, you know, there's like so much cool science that can be done. You know, but, you know, I feel like, you know, society is putting the brakes on us. And I'm like, stop, please. There's so much cool things that we can do.
No. Nina, thank you. And I think that's such a great articulation of the moment that we're in. There's a moment of stunning possibility, but there are these challenges. And for those that have questions or want to learn more, please talk to your contact in philanthropy. Somebody asked in the chat about how close we are. Please email Abby Norris at anorris2@fredhutch.org. And for those of you who want to learn more about Fred Hutch and the campaign for Fred Hutch, in this moment of stunning possibility, please go to fredhutch.org/campaign. Thank you all for joining. I really appreciate all your time. And thank you to our panelists, Nina, Kelsey, and Chris. Have a good afternoon.
Thanks. Bye, everybody.
Bye.
Thanks.